So now we know non-eukaryotic cells were one of the first to appear on Earth billions of years ago. Structure has proven that their structures and functions are adaptable. All bacterial cells have a cell membrane, cytoplasma, ribosomes, and few or less chromosomes. Almost half of them have a cell wall. There are a few particular structures found in some. They are flagella, pili, and that are just a few examples. With the bacterial cells. So, the bacterial cells look like they are two-dimensional. But when we look at them through a specific ordinary microscope, we will see there are different categories, that they are broken down to different groups, which is listed in the diagram. The first group is in all bacteria. We will find cytoplasmic cell membrane, bacterial chromosomes or nucleoid, ribosomes, and cytoplasm. In the other group that is listed in the diagram, we will find in some bacteria, the first is S layer, Fimbria, outer membrane, cell wall, actin cytoskeletons, pilus, capsules, inclusion, granule, bacterial microcompartments, plasma, and flagellum. And that is all the different groups. In the diagram on the right, it breaks down and tells you exactly what all of the groups does. Bacterial comes in all shapes and arrangements, like it's listed on the diagram. All shapes and sizes bacteria comes in, and they also reproduce. The average size is about 1 micron. So a lot of the bacterial species, each bacterial cell is fully able to carry out life activities. They can even reproduce and even form a metabolism. Some of the bacteria will work together in pairs or groups, and they will communicate through a certain chemical. And then that causes them to act differently than if they were single-celled. There are a few common shapes arrangements: the coccus, the rod, and the curve, which is demonstrated on the diagram. Genetic differences. If there is a genetic difference, it will cause variation in the cell wall structure. So what happens will be we will have a cell of Corneobacterium, which are normally rod-shaped, side by side, and they're somewhat considered club-shaped. So that means they display a lot of different shapes. The cells will either be considered single cells, a chain of many cells, streptobacteria. A lot of times the hinge will fold back and can form a side-by-side row of cells, which is demonstrated in the diagram. Now we have biofilms. So with everyday life, we have to deal with biofilms, and the certain ones are microbial habits. The reason is we have to deal with them is they have direct access to our foods that we eat, the water that we drink, and the atmosphere that we work or live at, or any of our atmospheres. Because what will happen is they will survive where oxygen level is the highest. And of course, we had to have oxygen to live. A lot of foods that we eat grow bacteria and fungi if we don't eat them in enough time. One of the main ones is pretty much the bread. Biofilms come in different waves of thickness, like demonstrated in the picture. It's dependent on their location of the growth, and it's even can become part of our mouth, depending on the time frame it's been since we flossed or brushed our teeth or had our teeth clean, which is pretty crazy because I didn't know that microbial could become part of our insides, like our mouth and teeth and stuff like that. We're next going to talk about flagella. So this is a wall the flagella can provide for the cells to swim freely. Just like when we go down a water slide, we swim freely outside of the water slide into the pool. That's the same thing that happens with these. There are 3 important parts to this. The filament, which you could see on the diagram, the hook, and the basal body. So what happens is the structure will have proteins that will insert to the curved tubular hook, and the hook will anchor to the cell by the basal body. Then we'll have the firm of the rings, when then they will anchor to the cell wall. It will begin to rotate as demonstrated. So that's pretty much like when we take a boat out. We're going to anchor it off in order for us to get off the boat or just to sit on the boat or whatever we want to do. It will anchor and it will come to a complete stop. So this one is about the magnetic process. I was surprised when I learned that some bacteria are magnetic because they contain a chemical of iron oxide, which allows them to have magnetic properties. So the bacteria will use these granules to use the polar and gravitational fields to pull the bacteria back and forth. And what that does, it causes for a deeper habitat to form. So that is pretty much demonstrated in the diagram. As you can see, the magnets are pulling back and forth. And I never knew that bacteria could be magnetic, but anything, I guess, can be magnetic. Vegetative is a spread that can determine what a species' shape or sizes are. You have the gram-positive and a gram-negative. What happens is they can form endospores, but the ones that we would be able to know and that are popular is the medically ones, and they are the gram-positive. And a lot of this makes sense because, or it's medically form of the bacteria can't spread and people can't get as sick easily because where the bacteria doesn't spread or grow, then it can't form a type of virus in order for us to get sick in the medically part of it. A lot of this is shown in a diagram as well, but if bacteria, like with the brass, bacteria will link to it, but it doesn't stay. So that's just like this. Bacteria can grow, it's just not going to spread because there's no way for it to spread. in this certain situation. The germination form, what happens is they would break a dormancy, and that will then begin to presence of water and specific chemicals or environment stimulus. And that is considered germination agent, which becomes present in the environment, as well as it varies among species. Usually small inorganic salts and amino acids when they will digest the enzymes by their membrane of endospores. They will then grow out of their coats and become fully active, which will then begin for them to go back to their vegetative cycle and start all over again. So it's pretty much like a chain reaction. It just keeps going and going and going. Archaea. This one has different parts to it. The first part of it will be what happens. It's considered the third cell type, which will be in a separate kingdom. It'll be under the domain Archaea. So they share many bacteria characteristics. They actually are near the domain Eukaryotic more than to the bacteria. The reason that is Archaea share a number of ribosome RNA, which a lot of them are not found in bacteria, and that is why the Archaea is closer to the eukarya more than it is closer to bacteria, just based on the more that they have in common. It doesn't mean that Archaea doesn't belong with bacteria, because some of the side effects, the Archaea will go with bacteria. It's just, it's more closer to the eukarya because of the more that they have to link up with. So this one is more about the Archaea as well. This Archaea is the extreme portion of it. It is the halophiles. In order for these to grow, they have to have a lot of salt, and even then they have high salt tolerance. So what that means is once they get the salt's going to multiply, and then that will form a sodium chloride solution. That sodium chloride solution is going to destroy the majority of the cells. They live in inland seas, salt lakes, salt mines, and salt ponds, and even in the Red Seas. What happens is you will not find these type of Archaea in oceans. The reason is the oceans does not have enough salt to perform for them to grow in order for them to survive. These halobacteria are responsible for the Red Seas. The reason that is the color of the red in the salt ponds is based on the pigments. And if the pigments is red, it gives off the color red from the pigments. So a lot of times we're not going to find this type of stuff in like Myrtle Beach or anything like that, because even though that the water in the oceans is salty, it's just not enough salt to order for these to grow. So they have to have a lot of salt, and which is demonstrated in this picture.